Welcome to Chapman Academy. This is a platform for you to grow and advance your research writing skill. I recommend you subscribe to Chapman Academy on YouTube, share with your friends and lovers so that they can also subscribe and join the movement as we all grow and advance our research writing skills. So I've been taking you through the various phases in clinical trials. Then today I'm going to teach you how to use the clinical trial phase 2 in your research writing. So in clinical trial phase 2, here we test to see how the intervention that we are coming up with is able to treat the intended outcome or the disease or the condition. That is what at phase 2 we test to see. In phase 1 we check for the safety and the efficacy, and then the phase 2 how the intervention is going to improve upon the life of the people. So in phase 2 of clinical trial, if the new treatment or the drug is found to be safe in phase 1, then there is a need for us to continue the phase 2. So after that we found out that the intervention or the treatment or the vaccine in phase 1 is proving to be safe and then promising, then we continue with the phase 2. Then we see how it will work with the intended outcome. So if it is cancer treatment, then we will try to see how it will work in the particular type of cancer we want to tackle, or the particular type of disease or condition we want to tackle. So that is what we do at phase 2 clinical trial. And then the benefit that researchers we expect to see depends on the particular intervention or treatment we want to provide. So if we are intending to suppress the spread of a particular condition or to take it away permanently, it depends on what we are expecting to achieve. So if it is a treatment that we are providing and we want to make sure that at the end of the day the condition should not exist again, then that will be the benefit that we are looking up to. So it is a cancer treatment and we want to treat the cancer once and for all without leaving any trace of the cancer cells there. Then it means that at the end of the treatment, we are going to see total completion or total diminish of the cancer cells. Or if we want to shrink or prevent the growth of it, it depends. So the benefit depends on what the research is about. And then in some study of phase 2 clinical trials, the benefit might also be to improve the quality of life, not only to treat a certain condition, but also to improve the quality of life and then make people get better. So if it is palliative care that we are providing, then it means that we want to make the livelihood of people better. We want to improve their living conditions, their quality of life. So let's say it is prosthetics, that's advanced prosthetics that we want to provide so that it can replace the missing part of the body. That one too is part of quality of life, providing quality rehabilitation support to people with affected disabilities. So these are what we look up to in phase 2 of clinical trials. And then in phase 2 of clinical trial, we recruit group from within 25 to 100 participants. So that's how many people we can recruit. And then we make sure that the people that we are recruiting, they are all within the same characteristics. So if it is cancer patients that we are recruiting, we need to make sure that all the patients are possessing the same type of cancer, or they are in the same phase or stage of a cancer disease. If it is hypertension, that hypertensive people that we are taking, we need to make sure that these people are all within the same type of hypertension. So that is how phase 2 is done. You need to make sure that the people that you are putting into your study, they are all within the same characteristics, so that you will be able to know and then measure. Because when you pick someone with stage 2 cancer, someone with stage 3, stage 4, you won't be able to do your measurement. You will be able to know the improvement that you are doing, because this person will be presenting different type or different amount of improvement, and then other people will be also presenting different things. So at the end of the day, it becomes very difficult for you to measure your results or your outcome, because you recruited people with different characteristics. So it becomes difficult for you, the researcher, to measure your outcome and then your impact on the patient. And then usually in phase 2 clinical trial, everyone gets the same dose. When we are giving 1.5 ml, everybody will get 1.5 ml of the drug. You need to make sure that you give everybody the same thing, so that if there is a change, if there is an improvement, you can account it to the treatment that you are giving to the people. But when you give this one 4.5 ml, give this one 3, you give this one 2, you won't be able to measure your differences, because the treatment is not given with a regular or with a standard measurement. So different people will be presenting different outcomes, and it makes your study difficult for you to measure and then know how your vaccine or your drug is being improving on the people's life, or how the drug is able to treat the particular disease, and then how best you can give the drug, because you are giving it on irregular basis. Although everybody get the same dose, but in some studies of clinical trial, people are randomly assigned to different treatments. They will give the same dose, but they will be assigned to different treatments. So maybe these people will be taking treatment A, and then the other group will be taking treatment B, but with the same dose. So if you are in treatment A group, it means that if everybody is taking 1.5 dose of the particular intervention, if it is drug or vaccine, everybody will take the same thing. And then if you are in group B and everybody is taking 5 ml of the intervention, everybody will take 5 ml. That is how it's done. You don't give different doses. Everybody take the same dose, so that it becomes very easy for you, and then know how your treatment or your vaccine is being improving the quality of life of the people. This group may get different doses or get the treatment in different ways to see which provide the best balance of safety and response. Like I said, although they are in different groups, but they get the same dose. If you are in treatment A, it means that you get the same dose, but maybe your dose might be different from those that they are on treatment B. So that is how it is. So that at the end of the day, we will be able to balance among treatment A and then treatment B, which of the treatment is best, and then safety, then is able to work to diminish the growth of the cancer cells or shrink the growth of the cells, or is able to treat the particular disease we want to tackle. So that is how in phase 2 clinical trial we want to see in phase 2? Because now we've tested for the safety. Now we want to know how the particular intervention is responding, is able to treat the particular disease that we are talking about. So that is how phase 2 clinical trial study is. And then mind you, in phase 2 clinical trial, we don't use placebo. We don't use placebo in phase 2. Placebo, we don't use it. I have a detailed lesson on placebo, so you can go to the videos, you get placebo. And then in phase 2, studies may be done at major research centers, communities, and then even the doctor's office or within the health facility. Phase 2 study can be conducted there. And then at this phase 2, large number of patients get the treatment in phase 2, and then we are able to identify less common side effects. Because here, when you recruit a lot of people into the study, you are able to identify side effects. Because let's say in phase 1 you recruit very few people, so sometimes side effects detecting become very difficult because it is few number of people, and then if those people do not present any side effects, you will not be able to detect less common side effect. But when you recruit more people, which is the phase 2 going, you will be able to detect less common side effects and then to give prompt attention to that. So you need to make sure that you recruit a lot of people, which is within 25 to 100 patients on the program, so that you will be able to detect the side effects. And then the key point of phase 2 clinical trial is that if enough patients benefit from the treatment and the side effects aren't too bad, then we will begin with the phase 3. So if we see that, oh, okay, a lot of people are enjoying our treatment and then we've not seen any bad side effects, and then the drug or the vaccine is being able to treat the condition, it means that we will continue with the next phase, which is the phase 3. And then in the treatment that have been shown to work in phase 2 clinical trial must succeed in one more phase. Yes, so if it works best in phase 2, then it means that it should be able to provide optimum benefit to the people in phase 3. It needs to prove itself positive in phase 3. It needs to prove itself positive in phase 3. It should be able to work best in phase 3 so that it can be approved. Because from the laboratory stage through to phase 2, and then it is still promising, then it should be able to prove itself positive in phase 3 so that it will be approved for the general community or the general population to have benefit of it. So that is what we do at phase 2 clinical trial. So in phase 2 clinical trial, in summary, we make sure that the drug or the intervention is able to work on the particular outcome we are looking for, is able to treat, is able to shrink, or is able to prevent the growth of the particular disease or the particular cells, cancer cells. So that is what we look at, and then we recruit from 25 to 100 participants. So that is what we do at phase 2 clinical trials. Thank you for being with Chapman Academy on YouTube. Subscribe to Sharpman Academy and you share with your friends and lovers so that they can also join the movement and learn new things every day on research. Subscribe to Sharpman Academy. Sharpman Academy. See you there.